I'm a cognitive neurologist who is interested in cognition and disorders of cognition in general, and specifically auditory cognition, which I'll explain as we go along. Although I'm working primarily on the brain, you might be able to see on my shelf a picture of the cochlea, and it's amazing how many disorders can be explained by going no further than the cochlea. But what I'm going to do today is consider links between cochlear function and dementia, which is something for which a number of models have been suggested. And in terms of the sort of worldwide impact of this problem, and who should care about this, the estimate here is 9% of 55 million. 9% is based on the figure developed by the Lancet Commission for the reversible contribution to dementia caused by hearing loss in midlife. 55 million is the current World Health Organization estimate of the worldwide dementia burden. And what I'm going to do today is take you through evidence for the link between hearing loss and dementia, think about some possible mechanisms, and I really want to develop a specific model which we're very interested in, that natural listening engages specific high-level cortical mechanisms that are susceptible to dementia, and particularly mechanisms in the medial temporal lobe that might interact with the Alzheimer's disease process. And in terms of the evidence for the link, this is actually quite good. There are 3 robust studies which were incorporated into the analysis and model developed by the Lancet Commission, in which subjects had their hearing measured and were followed up for periods of typically 10 years to look for the development of incident dementia. And this is one of the 3 studies, which was from the group of Frank Lin and colleagues from Johns Hopkins. And what the study shows is a link between the degree of hearing loss shown here on the abscissa and the hazard or increase in risk of dementia as a function of hearing loss. And you can see that there is a stratification of the risk, which increases as a function of the degree of hearing loss. And this risk is still present after you take account of other demographic factors, including age and a variety of vascular risk factors. What I'm going to do today is take you through 4 possible mechanisms for the link between hearing loss, based on, first of all, common pathology, which is affecting the cochlea and the cortex, an effect of impoverished input to the auditory cortex, increased listening effort, which occupies cognitive resources, and finally a specific interaction between the pathology of dementia and altered activity due to hearing loss. The models aren't mutually exclusive, I should say, and I've got a strong prejudice towards one of them, which will become obvious as the talk goes on. So this cartoon shows the first mechanism, and what you can see on the left are structures in the ascending pathway from the cochlea to cochlear nucleus, inferior colliculus, auditory thalamus, and thence to the primary auditory cortex, PAC. This is a coronal section through the temporal lobe, showing the auditory cortex at the top, and the medial temporal lobe structures, including the hippocampus in the midline. In terms of common pathology, Alzheimer's is well established as affecting the retina, which isn't surprising because the retina is actually part of the brain embryologically. But there are actually a number of genetic mouse models of Alzheimer's disease in which the disease process has been demonstrated in the cochlea in addition. Human postmortem studies have shown Alzheimer's pathology in the structures of the ascending pathway, the cochlear and inferior colliculus. But these are rather subtle changes, and in general, even gross lesions affecting those structures, like strokes, don't produce striking hearing symptoms. The other common pathological mechanism that might be relevant to links between hearing loss and dementia is vascular disease. And as you will all know, there are prominent models of vascular disease producing presbycusis, and vascular disease is well established as a cause of dementia. What I would point out, though, is that the majority of incident cases in the Lancet Commission analysis were actually Alzheimer's disease rather than vascular dementia. This is the second mechanism for this link, which is an effect of impoverished input to the auditory cortex. There's a large literature in animals that shows that enriched environments which stimulate the animals can improve the microscopic and macroscopic brain structure. And what's been proposed is that in humans, living in an enriched environment might increase your cognitive reserve because of this, and have a protective effect against dementia. If you have hearing loss, the argument is the reverse, that there is a decrease in the microscopic structure and macroscopic structure of the brain, which removes that protection from dementia. The third mechanism which we've considered is related to the idea that cognitive resources are limited. And this was first suggested by Danny Kahneman in the 1970s for working memory, and similar ideas followed for attention and language. And the idea is that that resources which are instantiated in high-level cortex might be used up by listening effort in difficult environments. I think this might be a model for cognitive deficits in the elderly with hearing impairment, as opposed to the specific disease that is dementia. But even then, I don't think it's a great model. And the reason I don't think it's a great model is because cognitive deficits in the elderly, with or without dementia, are still present when they are not listening in difficult environments. And this effect on listening effort would need to be simultaneous, I would argue. And this is the fourth mechanism, which I obviously have a prejudice towards. Mechanism 3 developed the idea that difficult listening engages brain resources well beyond the auditory cortex, including the medial temporal lobe. But mechanism 3 doesn't take into account the fact that the dementias all have specific molecular and cellular pathologies with particular distributions. And the idea here is that there might be an interaction between activity changes in the medial temporal lobe due to hearing loss and the pathology of dementia. So what's the evidence that natural listening engages these high-level cortical mechanisms beyond the auditory cortex in areas that are commonly affected by dementia? I hardly need to tell you all that cochlear function is not a good predictor of natural listening success, including speech and noise listening. Here are two subjects with very similar audiograms who get the same hearing aid. One develops speech and noise, which is good, and the other develops poor speech and noise and throws the hearing aid away. And there's a lot of evidence to show actually that listening to speech and noise in more natural situations depends on high-level cortex. This shows brain activity which is indexed by blood flow measured using PET during speech and noise listening. PET isn't used so much these days because it involves injecting radioactive tracers and has been superseded for a lot of normal studies by functional MRI. But we still use it quite a lot, actually, because it allows us to carry out functional imaging in cochlear implant users. And what these data show, these are sections, axial sections through the brain, which show activity in the auditory cortex whilst listening to speech and noise, but also consistent activity in individual subjects. There are 9 here, which is present in the inferior frontal lobes. If you look at the relationship between activity and performance in the auditory cortex and the inferior frontal lobe, it's quite interesting. This looks at the left auditory cortex and the left inferior frontal gyrus. And you can see that in the left auditory cortex, we see an increase in activity with performance, but actually we see the reverse in the inferior frontal cortex, which I think can be interpreted in terms of the activity in the inferior frontal gyrus, reflecting listening effort, which will actually decrease as you get better at the task. My group works on aspects of auditory cognition that are language-independent determinants of speech and noise, and we've already heard a little bit about our work on figure ground analysis, where subjects are required to separate foreground stimuli presented over tens of milliseconds from a noisy background. And we also work on auditory cognitive processes carried out over longer time periods that might be relevant to sentences. In terms of the first, this shows a stimulus that we've developed, which examines an aspect of auditory cognition relevant to speech and noise, which is figure ground analysis. And what you can see here, this is time on the x-axis and frequency on the y-axis. And this is a segment that shows the background where we have random tones in frequency time space. And what we can do when we carry out these tests is to constrain certain frequencies to remain on over a period of time. You can see that there are 4 here. And we've done a lot of psychophysics on this, but basically, if you have enough tones that are on at the same time and they're on for long enough, you hear a figure that pops out from the noisy background. So rather fancifully, we've called this an audiogram for scene analysis, the idea that it reflects something which is much closer to speech and noise listening than the pure tone audiogram. And we show in behavioral work that we can get prediction of actual speech and noise listening from this task. This shows the relationship between the actual audiogram and speech and noise listening in normal listeners. We're actually looking here at the high frequency audiogram between 4 and 8 kilohertz, and we can see a positive relationship here. But we can also see a relationship between the figure ground test, the detection task that I just showed you, and also a relationship between a discrimination task and actual speech and noise listening. And actually, when we carry out hierarchical regression, this demonstrates that the figure ground measures account for distinct variance in the speech and noise performance from that due to the audiogram. So we have separate processing, which is indexed by the figure ground measure, which I'm going to argue in a moment is due to brain processing. We've also looked at this in cochlear implant users, actually, and we get a stronger relationship between figure ground sensitivity and speech and noise listening. AZBio is an American sentence in noise measure. And when you do multiple linear regression here, we've come up with a model based on both the figure ground listening and indexes of peripheral processing based on spectral ripple and temporal modulation detection. And that model can explain about half the variance in speech and noise. So where does this happen in terms of the brain processing that's necessary for speech and noise? These are rather old functional MRI data which show activity during speech and noise listening, which is present not in the primary auditory cortex in the superior temporal plane. These are coronal sections through the brain, but actually out on the convexity of the brain in the superior temporal gyrus and the superior temporal sulcus on either side. And as well as doing functional MRI experiments on normal subjects, we can actually carry out invasive human neurophysiology on neurosurgical candidates, which is work that we carry out in collaboration with the neurosurgical group at Iowa University with my colleague Matt Howard there. And this example shows an electrode grid which is placed over the external surface of the temporal lobe. This cartoon shows the superior temporal gyrus, and in red you can see contacts where we show a significant increase in the activity measured by local field potentials that is present. And that's shown in these time frequency plots, which show an increase in activity in what is called the gamma range, high frequency oscillations, which occur after the figure comes on. And this is highly consistent, actually. We've looked at more than 12 subjects now over the last 6 years. The second memory mechanism that I want to consider that might be relevant to speech and noise is the grouping of elements over seconds dependent on working memory, holding sounds in mind. I'm talking here about your ability to hear to keep non-speech sounds in mind. I'm not talking about phonological working memory, although that has also been shown to show a relationship to speech and noise ability. And the idea here is that if you can keep sounds in mind over a period of seconds, it allows you to link together words in a sentence and might allow that sentence to pop out from the background. And what we've done here is to carry out experiments where we have measured your ability to keep a very simple sound in mind, just a pure tone. So subjects have played a pure tone, there is a delay of seconds, and then after that delay, they have to adjust a test tone to the same frequency as the tone they heard before the gap. Very simple. And what we do here is to measure the variance in the estimates that the subjects make, or precision, if you like, as an index of how good their working memory is. These are data from my colleague, Maya Ladd, who is a neurologist who works on this with me, showing a relationship between your working memory ability and speech and noise analysis. And we get a nice relationship here, which has been replicated and shown even greater effect size in a much larger population. We've looked at the brain basis for this as well, in a paradigm where subjects are required to keep tones in mind. They hear 2 tones, they're cued to remember 1. Then after a delay, there's a probe tone, which they have to decide whether it's matched or not to the previous tones. And we see activity here in auditory cortex during the retention phase of working memory, measured in this way. And we also see activity in the inferior frontal gyrus. So it's very similar to the activity I was showing you during speech and noise listening earlier. But what we also find here, which was a bit of a surprise to us, is that we've seen activity which is present in the hippocampus, in the medial temporal lobe, during the period of retention for simple tones. And the hippocampus is not well recognized as a substrate for working memory. It's much better recognized as a substrate for episodic memory, as described in the in a large body of neuropsychological literature. And we've also looked at this with intracranial recordings, which we've carried out in subjects from the auditory cortex in the top 3 panels here, and also in the medial temporal lobe structures in the bottom 2 panels here. At the top, you can see in the auditory cortex responses to the initial 2 tones, and this is the probe tone after the delay. And I want you to focus here on the medial temporal lobe, where you can see low frequency activity below 10 hertz in what is called the theta and gamma range during the delay period in both the parahippocampal gyrus and the hippocampus. And this was highly consistent actually across 6 subjects. You can see the increases in low frequency activity in those 6 subjects in hippocampus and parahippocampal gyrus here. So the short version is that speech and noise perception itself, but also aspects of auditory cognition that might predict speech and noise function, both depend on highly distributed mechanisms that are beyond what is traditionally regarded as auditory cortex. And there is further evidence too. This shows changes in the structure of a brain, of the brain over time as a function of the degree of hearing loss. This is a coronal section through the brain. This is the temporal lobe of the brain again. Basically, the darker the blue, the greater the decrease as a function of hearing loss. And what I find really interesting in this study is that the greatest changes that are due to hearing loss are seen not in the auditory cortex, but in the medial temporal lobe. This shows an experiment I did 10 years ago now, where we looked at the reverse process, not loss of hearing, but increase in hearing ability. And this was a study we did on piano tuners, where we looked at the grey matter density, the amount of grey matter, if you like, as a function of prolonged specialized listening in piano tuners. And what we saw here is a clear relationship between the grey matter density in the hippocampus on both sides and the years of piano tuning experience. Moving on from humans, these are rodent data, which has actually been carried out in an experiment where normal rats listen to a tone sweep after pressing a bar, and they're required to release the bar when the tone sweep gets to a particular frequency. What was found here is that units in the hippocampus were demonstrated that were tuned to a particular point in frequency space that was part of the sweep. And they only showed this activity when there was an active task. You didn't see it during passive playback. What was particularly interesting is that a lot of these units were also what are called place cells, which are active when rats are in a particular part of physical space when they walk around a room. So the idea here is that the computational mechanisms in the medial temporal lobe that are well established for spatial analysis might also subserve the analysis of sound constructs. And we've actually started in Iowa now to actually record from single units. This shows which we can do rarely using a specialized electrode arrangement that we can use. This shows a recording from the hippocampus where we can record single units. And this shows that we can show tuning of that unit to a particular point within what you might think of as an acoustic soundscape. Basically, it's a type of patterned noise with different degrees of granularity. So very briefly to finish, I'm going to fly a kite and suggest that there might be interactions between the disease process that is responsible for Alzheimer's disease, which typically starts in the medial temporal lobe in the transentorhinal cortex in this classical study. And the idea is that increased activity in the medial temporal lobe, which is present when subjects carry out exhibit greater listening effort, might interact with the disease process that is Alzheimer's disease. And I've developed a model for this, which is based on Alzheimer's disease pathology causing an increase in activity and excitotoxic cell death in the medial temporal lobe, which is compounded by further increases of activity due to hearing loss. But there's another possibility too, to unfly my kite, which is that in fact the critical aspect of decrease of hearing loss causing dementia might be decreased activity in the medial temporal lobe, which might be protective against Alzheimer's disease. And these are rodent data from a group in Caltech showing basically that repetitive acoustic stimulation can lead to an improvement both in the behavioral changes in an Alzheimer's model, but also in the deposition of amyloid and phosphorylated tau in the auditory cortex and in the medial temporal lobe. So I'm going to finish now with some clinical comments, just to say that the established link between hearing loss and dementia is yet another reason to treat hearing loss aggressively in midlife. I really don't like the term reversible, which the Lancet Commission have used, because I think it might be irreversible, but it might be preventable. We need better data to establish if auditory cognitive measures beyond the audiogram might predict dementia better than the audiogram itself. And I think we also need better data to establish links between hearing loss and different molecular subtypes of dementia beyond Alzheimer's disease. So I'll finish there. I apologize for overrunning a bit, and just to acknowledge the large number of people who have contributed to this work, including your next speaker, Will Sedley. Thank you.